So it is my great pleasure to introduce Dr. Brian Kim. And Dr. Kim received his MD degree from University of Washington. And then he did a residency and a postdoctoral training in the University of Pennsylvania. And then in 2014, Dr. Kim was recruited to WashU in the Department of Division of the Dermatology and the Department of Medicine, right? So as assistant professor, also the co-director Center for the Study of Itch. So for that many years, and he's he had a lot of achievements and published very high-profile papers, and particularly this research focused on the immuno mechanism of the skin disease and also sensory neuron disorders, and including the itch. And so he just published a Cell paper and a number of others. And so I'm going to be short just to say, and it's a great pleasure to have you to share your results and your achievements. And the title of your talk is shown here, and is the neuroimmune regulation of itch. Thank you. It's all yours. Yes, it worked.
Unmute yourself.
You're mute.
You're still muted.
Sorry, my bar was hiding from me. Let's go back here. All right. Well, thank you, Shiming, for the invitation. Thanks to all of you. Hopefully this will be interesting. It will bring some new insight into both biology and clinical problems that I think there's some relevance here, actually, particularly to the conjunctival disorders and itch, which I think plague a number of patients. We'll just get right into it. I have a number of disclosures that I'd like to highlight, ranging from consultancies to grants and patents. Happy to discuss any of these if anyone has interest. The central focus of my laboratory, my program has been for over 10 years now, is understanding how does healthy skin become inflamed skin? And more recently, how does inflamed skin result in itch? And what are the molecular and cellular mechanisms that regulate this? I think this is actually a very broad question because you can apply these principles to almost any barrier surface or organ to understand essentially chronic inflammation. We've used this disease, it's called atopic dermatitis or eczema. Many of you will be familiar with it. Predominantly affects children. It's in the family of atopic disorders. We often refer to the atopic triad with shared underlying physiology as food allergy, asthma, and eczema. This also is a part of the family, though we don't talk about as much, is allergic rhinitis and allergic conjunctivitis. The most important point here is that itch is the central symptom of the disorder, and this is something we've been tackling more recently. But when we first embarked on these studies, I have to admit, we weren't really focused necessarily at that time on the mechanisms that trigger itch, but we're simply understanding of what results in rash. And the paradigm at that time was that you have environmental stimuli or allergens that somehow translocate through a damaged barrier, and then that results in the activation of adaptive T cells, which recognize antigen. To this day, we don't know what antigens actually drive this disease, or if any. But notwithstanding that, these adaptive T helper type 2 or Th2 cells, through the production of these Th2 cell, or now we refer to as type 2 cytokines, drive the production of a variety of different pathologic processes, including production of IgE, activation of mast cells, which are coded by IgE, which can recognize allergen, and by way of these cytokines, direct damage to the barrier. During my postdoctoral fellowship, I wanted to look at this disease in a very different way. What really inspired this was the discovery of the epithelial cell-derived cytokines, IL-25, IL-33, and TSLP. What was intriguing about these cytokines was that they are released from stressed epithelia, and is exactly what we see every day in the clinic, is stressed epithelia, and was known at that time that broadly this allergic inflammatory axis is activated by these cytokines. What kind of started my career in this field was that in my postdoctoral fellowship, we then discovered rare circulating basophils, which get recruited into the skin as major sources of IL-4. And then we discovered the first time group 2 innate lymphoid cells, or ILC2s, in the skin, and that these were actually major sources of IL-5 and 13, shifting the paradigm a bit away from the antigen-specific adaptive T cell, and now being much more inclusive of the possibility that these innate cells are actually major sources of these type 2 cytokines. And what I would argue is that subsequently that's been proven not just in the skin, but across a number of barrier surfaces now. Now, blocking IL-4 and IL-13 has become a major therapeutic strategy. It would not be an understatement to say it's revolutionized the treatment of atopic dermatitis by way of the drug dupilumab, which blocks IL-4 and 13 at the receptor level, IL-4 receptor alpha. Actually, I have been collaborating a bit with Dr. Margulis and Dr. Wong about some of the complications of this, some of the ophthalmologic complications. In the interest of time, I'll get into it now. But this, notwithstanding that, is a very effective and safe therapy for moderate to severe atopic dermatitis. At the time that this drug was being developed, there was a lot of buzz about this kind of unprecedented effect that was being observed in terms of the anti-itch effects of these therapies. And we were already exploring how these cytokines might actually engage the nervous system. So to talk about the nervous system a little bit, and I think many in the audience may be more familiar with some of the, particularly the autonomic physiology, which I was more familiar with prior to going into this field. But with the simple way to think about the skin is that there are two kinds of peripheral nervous systems. There's the autonomic nervous system, which we know it regulates everything from heart rate to breathing, and in the skin, regulates vascular tone, which is not exclusive to the skin. A unique feature about the skin, though, is that there are actually not just adrenergic sympathetic nerve fibers, but there actually are cholinergic sympathetic nerve fibers, which cholinergic is usually attributed to the parasympathetic, that regulates sweating. I don't mean to be facetious, but it seems to me this is pretty much all we know about the autonomic nervous system in the skin. There's very little known about this. Now, what we study is a somatosensory nervous system, which essentially allows us to consciously perceive the outside world, whether this is through a variety of different fibers. A beta fibers, can detect friction. A delta fibers, which can detect other mechanosensation as well as pain. And then C fibers, which house a whole variety of sensations, including pain, itch, temperature, et cetera, et cetera. And of course, a lot of the sensations that mediate in the skin are also mediated in the conjunctive as well by this nervous system. What's interesting about the somatosensory nervous system is that there are only a few parts of the body that this was really designed for. The somatosensory nervous system does not innervate formally the gut or the lung or any viscera. It's for somatosensation, so it's really kind of everywhere you can see on the body is pretty much where that's innervated. If we're going to talk about the nervous system, just to put everyone on the same page, what do we mean by the nervous system and neurotransmission? Well, when we think about your typical neurotransmitter, it binds a G protein coupled receptor on the terminal axon, typically, and then this results in the opening of cation channels like TRP channels, which I'll talk about in a bit. You get cations crossing the gradient, and what happens is that with sufficient depolarization, what will happen is you'll trigger then these voltage-gated sodium channels to open. They'll flood in sodium, and you'll get an action potential. And you'll get an electrical signal to the spinal cord. The spinal cord will relay up to the brain, and then you will perceive itch, pain, or whatever sensation. So how do we bring this now more specifically back to some of the biology and itch? Well, let's start with histamine. We're all familiar with histamine. Everyone's probably taken an H1R antagonist. Well, how does histamine work? You inject it into the skin. It binds its receptor, H1R. It will trigger an action potential, and you will sense itch. This is almost entirely dependent on having a functional TRPV1 cation channel here, which responds to the hot pepper ingredient capsaicin. Well, one of the issues, with histamine has been that antihistamines have been, although very effective in certain kinds of itch, has been woefully lacking in efficacy for the vast majority of chronic itch disorders in the skin, also atopic dermatitis. And this has always been something that's really nagged us for many years. But new hope came by way of the cytokine IL-31, which acts as a neurotransmitter was originally reported to be predominantly derived from that adaptive Th2 cell I described earlier, binds its receptor on sensory neurons, and essentially acts exactly like a neurotransmitter and can trigger itch with variable dependence on other TRP channels like TRPA1 and TRPP1. So what this told us that maybe we don't need to focus so much on histamine. Maybe there are other pro-inflammatory factors that may act as neurotransmitters that we can disrupt for itch. And in fact, clinically, this has actually emerged as indeed a therapeutic strategy with a drug called nemolizumab that's in development, which is an IL-13 receptor antagonist antibody. Its primary efficacy derives from its anti-itch property in atopic dermatitis. It is also now shown efficacy in another primary chronic itch disorder called prurigo nodularis, in which the itch arises primarily from the nerves rather than from the inflammation in the skin, so slightly different way at which you arrive at the itch. And so this is clearly now an emerging therapeutic strategy. This is what prurigo nodularis is, in case you're wondering. It's slightly different from eczema. You actually get these kind of dome-shaped hyperkeratotic papules. Again, it's largely thought to be secondary to a lot of the itch, which results in the individual rubbing and scratching in a manner to cause these secondary lesions. Well, taking a note from what IL-31 does, in recent years, inserting our biology into this, we came to ask a very simple question, which is, do these canonical or classical type 2 cytokines activate the sensory nervous system in a similar manner? To test this, my first graduate student, who's now a resident in neurology at Harvard, Landon Etchen, discovered that by extracting dorsal root ganglia, which house the sensory neurons, peripheral sensory neurons, and just simply looked at expression of the receptors for these type 2 cytokines. And he found that, in fact, the receptor for IL-4 and 13, IL-4 receptor alpha here, was detectable. We could never detect IL-5, and we could also detect the native IL-13 receptor alpha-1, which signals IL-13, as well as IL-31 receptor, as you would predict. What's interesting is that from cadaveric human donor DRG, this expression pattern is highly conserved. We actually think that there's a lot of functional homology as well. I'll show you that here. So we know that these receptors we knew that these receptors are expressed now in the sensory neuronal compartment. The question then is this functionally important? Do they even respond to these cytokines? We can test this by doing calcium imaging. So on the y-axis, we look at the fluorescence. We can actually detect by fluorescence calcium influx into a neuron over time in the x-axis. And if we spike in recombinant murine IL-4, what we find is this nice spike. We can confirm by way of potassium chloride that these are in fact responsive neurons that are healthy. We can do this with human cadaveric DRG as well. We can spike in recombinant human IL-4, and we see this nice spike within a few minutes. Here, we note that these neurons actually responsive to capsaicin, indicating that they are actually TRPV1 positive, which about 75% of the sensory neuronal compartment should be TRPV1 positive. So the question then is, neuronal restricted expression of these receptors important? So sure, they express it, they respond to cytokine, but who cares? So we use mouse models to test this, to preclinically validate whether this is in fact what we could predict happens in patients. To do this, we actually use a validated mouse model in which we use a topical irritant called MC903 on the skin. These mice then very reliably develop AD-like disease or atopic dermatitis-like disease clinically, scaly red skin, histologically inflammatory inflammation that we can grade, as well as immunologically, we can do flow cytometry and look at their immunological composition, which very much mirrors what's been reported in human atopic dermatitis. If we do RNA sequencing in the skin, what we find is that by just bulk RNA sequencing, we essentially see the who's who upregulated of cytokines, chemokines, keratin alteration in the skin as reported in human atopic dermatitis lesional skin. Importantly for us, in terms of itch, we can measure scratching bouts in these mice, and what we find is that over time on the x-axis in days, and scratching bouts on the y-axis, numerically, these mice are exhibiting very robust spontaneous scratching, so they actually exhibit chronic spontaneous itch. I want to underscore that they have chronic spontaneous itch, and what I mean by that is at any given moment, as these mice develop the disease, if you look at these mice, you can tell that they are scratching very robustly. That's in contrast to evoked itch, in which you say you inject histamine, the mouse itches, it goes away, and it's not until you inject histamine again that the mouse itches. So this is a very disease anchored chronic spontaneous itch, and it'll be hopefully apparent why I'm stressing that point. So now in vivo, using this model, we can test, is neuronal restricted IL-4 receptor alpha expression critical, and is this important at all physiologically? So we then genetically deleted in conditional manner IL-4 receptor alpha just on peripheral sensory neurons. We're using a sodium channel Cre recombinase, and then we could run this mouse through the model, and what we find is that indeed there's a marked significant reduction in terms of itching, over time, or scratching, here shown on the left. That's also in conjunction with a reduction in terms of the skin thickness, as well as the inflammatory infiltrate upon histology. So in the interest of time, this work is published. I'm not going to get into the nitty-gritty details, but what we found is that how does this work? So IL-4 and IL-13, they're not really pruritogens. In other words, they don't cause a lot of itch if you inject them into the skin. But what they do is they dial up the neuronal sensitivity and allow the neuron to become much, much more sensitive to other pruritogens. So it essentially, drugs like dupilumab, what we think it does is that it actually anesthetizes the nerve to some degree in terms of its ability to respond to a variety of other itch-inducing pruritogens. But what's also interesting is that in recent years, the neural circuitry of it has been unveiled by a number of people, many in our center like Yufeng Chen and Chen Lu. And what's been essentially codified now based on single cell RNA sequencing is that there are different subsets of itch sensory neurons. Some that are MRGPRD positive, some that are MRGPRA3 positive. These are itch receptors. Some that are IL-31 receptor positive. And they're very different and they're distinct. But what was interesting that Landon observed early on is that all of these itch sensory subsets actually respond to IL-4 and 13, indicating that perhaps actually there may be a broader anti-itch kind of effect of drugs like dupilumab. And in fact, dupilumab is being investigated for the other condition called prurigo nodularis, and this clinical trial will read out this year. And we've actually found that also anecdotally in case series that patients with idiopathic forms of pruritus or chronic pruritus of unknown origin also respond quite well to dupilumab. What do I mean by chronic pruritus of unknown origin? This is actually a distinct form of itch. It's what I see actually most in the clinic. So in contrast to atopic dermatitis, in which you see very clear lesions in the skin, these individuals actually have almost no rash. They'll often just have scratch marks. It develops later in life. It's quite maddening because they have no real history of itching or atopic dermatitis. So here's an example, another example. These patients will just have simply scratch marks here on the upper back where this individual cannot reach. His skin is clear. So this is not a primary process, this is a secondary process. And then down here where he can reach again, he starts to develop a rash on the upper arms. Following treatment with dupilumab, you see the scratch marks simply go away. Again, very anecdotal. This is an experience with, I think, about 15 patients here over a long period of time. These are patients that we just followed in the clinic who were able to get dupilumab off label. And what we found is that these patients, starting off with a very severe itch, so 10 is your worst imaginable itch, 0 being nothing, 7 or above, so severe itch, you can just see how these patients improve over time. That's somewhat of a variable response, but some of these individuals respond very, very robustly. Great, IL-4, IL-13, dupilumab's out, but at that time, dupilumab was actually not approved. And what we were interested in is there another way downstream of these cytokines that we could choke off either IL-4, 13, or even other cytokines that have not been discovered yet to be involved in itch by choking off these pathways? And as an immunologist, we know that these cytokines have to signal through their cytokine receptors and downstream JAK-STAT signaling. So we thought, okay, what if we just go after JAK and then we choke off whatever cytokine it might be? And so if we go back to the single cell RNA sequencing data sets shown on here on the right, we actually looked for expression of JAK, and what we found that it was JAK1 was enriched within these pruriceptive or itch receptive neurons somewhat selectively compared to other pain receptive or mechanoreceptive neurons. We did not detect any of the other JAKs. So what we actually found was that, in fact, we took a very similar approach in mice, and what we now believe is happening is that these cytokines and the JAK signaling, we know them to be very important for pro-inflammatory processes in the immune cell compartment. But what we're now proposing is that there's actually a neuromodulatory property that we can take advantage of, whether it's dupilumab or JAK inhibitors. And I'm not showing the mouse data because it's published, but we proved that even conditional deletion in JAK1 is sufficient to ameliorate itch, much like with dupilumab, and actually much more potent. But also we were able to get patients the pan-JAK inhibitor, so JAK1, 2, 3 inhibitor, tofacitinib on patients with this chronic pruritus of unknown origin. And again, very anecdotally, we found that in case series that these patients improve. You see their scratch marks go away over time following treatment, and as well as their itch score. So what we actually think is there are many, many chronic itch disorders that we've known about for a long time that have been largely ignored in the clinical realm. And the way we're thinking about this is that there are certainly ways that you can get rid of itch, as in atopic dermatitis or an insect bite or scabies, where if you get rid of the infestation, you get rid of the inflammation, and you can get rid of the itch. In other words, if you treat someone with atopic dermatitis with high-dose prednisone, you can get rid of the itch. Not an ideal strategy, but you can. But then you have other forms of itch or very neurogenic in origin. In other words, the itch is starting in the nerves, where anti-inflammatory agents are not going to give you much mileage, and you have to use neuromodulatory agents. Well, some of these agents that were originally conceived to be anti-inflammatory, we think actually have a very dual properties. But then, so how does this work then? We think that there could actually be efficacy even in the realm of some of these neurogenic disorders, because now we're discovering a lot of these neuroimmune axes that work directly on the nerve. And there are conditions that sit kind of in the middle. So you have these conditions like chronic pruritus of unknown origin or prurigo nodularis, where there is some inflammation, but not a lot. But there's also neurodysfunction, not a lot. It's hard to know what came first, but there's really a good mix of the two. And the idea is that you want a two-pronged approach of both anti-inflammatory as well as a neuromodulatory agents. If we bring this back to atopic dermatitis, do we see some of the biology playing out in patients that we predicted? At this time, when we were putting a lot of the work together, we shared our data with Incyte Corporation, who actually had ruxolitinib cream, which is a JAK1/2 inhibitor, and we designed a phase 2 dose-ranging clinical trial for atopic dermatitis with this cream. And we designed it in mind with really looking carefully at itch. We enrolled 307 patients over 6 groups: vehicle control, so placebo control; active control comparator, so mid-potency triamcinolone 0.1% cream steroid, essentially the standard of care for atopic dermatitis, and a wide range of doses of ruxolitinib over 8 weeks was essentially the trial design. This work is published, so I'm just going to highlight the main points here. If on the y-axis you see a mean change from baseline in terms of the itch score, what we saw very early on, within 36 hours, was there was a significant reduction in the itch in response to the highest doses of ruxolitinib. That was already we could see trending towards beating out triamcinolone, the active comparator, and certainly already beating out placebo, indicating with this kind of rapidity that this may actually be a neuromodulatory effect of the topical ruxolitinib. The phase 3 trials have gone brilliantly, and they've actually shown anti-itch effects within 12 hours. This drug will, I think, no doubt be approved this year. Probably my prediction is sometime in the summer. It'll be the first FDA-approved topical JAK inhibitor for atopic dermatitis. So just hot off the press here, actually today, this is a JAK1 selective inhibitor. This is an oral inhibitor by Pfizer called abrocitinib. And they compared this to placebo, but also dupilumab, the monoclonal against the IL-4 receptor alpha. And we predicted in our mouse work that what would happen is that, given that so many more cytokines can signal through these JAK-STAT pathways, that actually choking off JAK would have a broader and maybe much more potent anti-itch effect. That's at least what we saw in mice. Did this play out? Yes. If you look at the highest doses of abrocitinib, we see the probability of a 4-point improvement on this itch scale, beating out dupilumab, shown here in the green. So orange is beating out the green. So we think a lot of the biology that we unveiled is playing out in clinical trials. And 2021 is going to see probably 4 or 5 JAK inhibitors approved. So it'll be very exciting to see how this all plays out in the real world. The blockade of these pathways, whether they be the cytokines or the JAKs, is one of the most active areas in the whole field of dermatology. It's absolutely explosive. These are just some of the highlights of some of the trials just in the last couple of years. So we'll see a lot happening clinically. So this brings a lot of great hope to our patients. Okay, so for the next part of the talk, I'm going to take a little bit of a different direction here. I'm going to talk about work from dermatologist and physician scientist postdoc Feng Wang in my lab. And as we were seeing all these clinical trials for atopic dermatitis come out and some of the trials that I was designing, one of the things that we were lamenting is the fact that so much of these endpoints and readouts in clinical trials get regressed back to a mean. So you can never really look at particular subset data. You can't capture certain things if that aren't necessarily in the design of a study very well. And one of the things that we do know, patients with atopic dermatitis, is that sure, they have chronic itch, but they also get superimposed acute itch flares, often triggered by some kind of environmental exposure. We don't necessarily know what they are, but as an example, you have a patient, a young child who goes to his grandmother's house, and every time he goes to the grandmother's house, there's a cat there and he itches like crazy. So what's going on there? This is not captured in clinical trials. It's not really measured. But what we did was to get at this a little bit more deeply, we actually collaborated with Regeneron Corporation, which developed dupilumab. And they had actually high resolution itch data, at least in the first couple months of patients in these trials. We asked for the data. They were actually gracious enough to collaborate with us in this manner. And they're able to get data with it, daily itch score data. We were able to get from patients just from the placebo group, so untreated, just placebo in these phase 3 trials. So no itch to worst itch, 0 to 10 scale, daily looking at high resolution itch. So how does this play out? Well, you see patient 1 here. This is the acute itch I'm talking about. He or she will start off with an itch score of 4, which is in the moderate range. It'll jump to an 8, come down to a 5, go up to a 7, come down to a 4. It's immediately intuitive if you see what's going on in terms of the lability of the itch in this individual that this is probably very debilitating. You see the patient in blue who has a moderate itch score of 5, roughly would average out to roughly the same as patient 1, but you see that he or she has itch that's fairly stable over time. It's not really changing. So then the question becomes, who has it worse? I would say the patient in red. What's going on with the patient in red? Why does this individual have such a different pattern of itch? And that was the question we sought to ask. And in fact, if we look over 159 of these patients, about a quarter of them actually exhibit this acute itch flare pattern while the others do not. So the question then we asked is this actually a subset or is this something that changes over time? We actually had hypothesized that this is probably something that everyone would experience if given the right environmental exposure. And what we found is that we start off with these patients in this kind of candy cane pattern here. In the first month, if we look in the second month, a small proportion of these individuals lose this acute itch flare pattern. Then in the bright red, we see that that patients who were classified as not having this acute itch flare pattern actually acquire this pattern. So again, suggesting that this is a dynamic thing. It's not necessarily that only certain individuals experience this, but over time that many individuals with atopic dermatitis may actually experience this kind of pattern. What am I really getting at here? Well, we study type 2 immunity. We essentially study what we think of as allergy or atopy. And atopy is defined as the tendency to produce an exaggerated IgE immune response to otherwise harmless environmental substances. But this is where it's kind of interesting. We know, so if you have allergic conjunctivitis, allergic rhinitis, you know that IgE binds allergens, and seasonally, if those allergens you're exposed to, the mast cells will degranulate, you'll itch, you'll get inflamed, you'll get mucus secretion. We know that if you have food allergy and you have allergy to peanuts, if you taste the peanut, you could get anaphylactic because your mast cells explode by way of IgE and you could die. Asthma, we know subsets respond to aero allergens in a similar manner. However, atopic dermatitis, despite being a bona fide atopic disorder, we've known for decades that these patients harbor allergen-specific IgE, but it's been unclear what they do. It's highly controversial that they have anything to do with the rash itself. And so we thought maybe this is what's going on. Maybe it's this reactivity, this acute reactivity is actually what's driving more of this itch pattern than anything else. And in fact, what we then did was we actually looked at these patients for these clinical trials with Regeneron. They were profiled for the serum allergen-specific IgE. And we just bin these patients into those who actually harbor allergen-specific IgE that's detectable and those that don't. And then we see that the percentage with acute itch flares is much more enriched in the individuals who actually have and harbor allergen-specific IgE in their sera. So this is where we wanted to take a more mechanistic approach. We went back into the mouse model. I introduced this earlier. What we do is we topically apply this compound called MC903 on the skin. And these mice, as I mentioned earlier, develop chronic spontaneous itch associated with lesional atopic dermatitis-like inflammation in the skin. But what's interesting is there's a different spin here. We can actually topically apply simultaneously ovalbumin, or OVA, as a model allergen, and these mice get sensitized to OVA. If you now intradermally inject OVA back into the skin, not even in the lesional skin, just in the cheek skin, just lateral to the lesions, these mice will develop robust acute itch flares, and I'll show that in a second. Okay, what happens to control mice? If they're vehicle treated, they get no rash, they get no itch, no surprise. Because they have no rash, they have intact skin, it's not leaky, you try to sensitize to OVA, nothing happens. So now when you intradermally inject with OVA, there's no hypersensitivity reaction whatsoever in these mice. Okay, so do they have allergen-specific IgE or OVA-specific IgE? Yes, the MC903 confers this sensitization to OVA, so they harbor it, so they are able to react by way of IgE to OVA. Now, what happens in terms of the itch? Let's start with the control mice because they're straightforward. Open circles, blue here. Scratching bouts on the y-axis, time intervals on the x-axis. You see these mice, they're not itchy. There's no chronic spontaneous itch. They don't get sensitized. So if you even challenge them here with OVA, nothing happens. They're fine. They're going about their business. Now let's get to the closed blue boxes here. Chronic spontaneously itchy. So baseline, they're itchy. They get sensitized to OVA. You challenge them and they spike. Over the course of about 30 minutes, the itch starts to resolve. So it's indeed an acute itch flare. And it goes back down to baseline chronic spontaneous itch. If you look in the open squares here, these mice have chronic spontaneous itch. They got MC903. They also got OVA, so they're sensitized. But now you challenge it with irrelevant bovine serum albumin, nothing happens. Because they have IgE to OVA, but they don't have IgE to BSA, so they're fine. So this is the model. So this is an acute on chronic itch flare model. So as I mentioned earlier, the role of IgE in atopic dermatitis has been very unclear. And so if we take IgE deficient mice, there's no effect on the chronic spontaneous itch. So I apologize, my mouse is finicky. No effect on the chronic spontaneous itch whatsoever. However, on the right, with the acute itch flares, we see the abrogation of this acute itch flare in IgE deficient mice. So that's not surprising. I mean, we should test it, but it wasn't surprising to us. Now what if you just don't have mast cells? If you have c-Kit knockout mice that development and lack mast cells, no effect on a chronic spontaneous itch. Not surprising, the role of mast cells in atopic dermatitis itself has been very controversial. Here's where it gets surprising. We get rid of the mast cells, no effect on the acute itch either. That was surprising because the mast cells are thought to be the primary responders to IgE. So we thought, okay, mast cells are tissue resident. They release histamine, they bind IgE, they're the primary responders. But there's another cell we've been studying for a long time now called the basophil, which is a rare circulating granulocyte, can go into tissues, also harbors a high affinity receptor for IgE, releases histamine and other molecules, but again, it's circulating in a much less common population in the body, but it can respond. And so we suspected maybe basophils are at play here. So we went back into patients. If we now draw blood from patients, look at basophils, and do they have a different phenotype? Well, a canonical basophil activation marker is CD203C. And we see that, in fact, there's upregulation of CD203C by flow cytometry, enumerated and quantified on the right. And additionally, importantly, if we look at the high affinity IgE receptor, Fc epsilon receptor alpha, we see upregulation of that Fc epsilon receptor, also quantified on the right. So what this tells us is that chronic inflammation associated with atopic dermatitis makes the basophils angry and poises them to be much more responsive to IgE. If we go back into the mouse model, we find that mouse basophil activation marker CD200R also is upregulated, as well as mouse Fc epsilon receptor as well, indicating very similar physiology. So up to this point, it's never been shown that basophils, these are rare, when I was in medical school, I was taught that they're vestigial cells of essentially no physiologic relevance. But when no one had shown up to this point that basophils alone are important or are sufficient to cause itch. And so we took a interesting kind of chemogenetic approach just as a proof of concept to say, okay, basophils are important. We took basophil specific MCPT8 cream mice and we crossed them to these designer receptor mice in which we could actually then conditionally insert a designer G protein coupled receptor just into basophils. That allows us to chemogenetically activate only basophils in vivo. How does that work? We inject clozapine. Clozapine selectively binds this G protein coupled receptor. It's a relatively inert pharmacologic compound, doesn't really do much else. So then what happens is the basophils explode, and then you can see what happens. In vitro, we find that the basophils actually marked by green here, and then we stain with avidin and have this degranulated pattern once we kind of expose them to the clozapine or CNO. And importantly, with one injection systemically of CNO or clozapine into these mice, we see that they develop an acute itch pattern, indicating that in fact, basophil activation alone is sufficient to cause itch.
Excuse me, could I ask you a question real quick?
Absolutely.
What exactly is MC903, and is it specific for basophils? Is that why you're getting basophil activation?
No, no. MC903 is calcipotriol. It's a vitamin D analog, and it's really just it's an irritant model. What it does is that this vitamin D analog in the keratinocytes upregulates expression of a lot of these pro-inflammatory cytokines, and it sets the mouse off the skin into a cascade of inflammation in the skin that mirrors kind of the models what we see in human atopic dermatitis. Does that make sense?
Sure, yes, fine.
Yeah. So the MC903 doesn't bind basophils itself. A key cytokine that does activate basophils that's induced by the MC903 is TSLP. And TSLP is induced in keratinocytes transcriptionally by this vitamin D analog. So it's, again, just really a model. There's lots of ways you can get to atopic dermatitis. Once you're there, though, there are a lot of things going on. The question is, what do you block to kind of intervene? So now here, what we're trying to test is whether basophils are required for this acute itch flare model that we've now developed. So we have this chronic spontaneous itch. Now we can challenge with OVA to elicit this acute itch flare, and we can actually deplete basophils in the special line called the Bastrec mice, in which mice only express the diphtheria toxin receptor on basophils. And so by way of diphtheria toxin, we can actually then deplete basophils, shown here as the CD49B Fc epsilon receptor compartment. And you see that there, with the depletion of these basophils, we see this amelioration of this acute itch flare. So the question then is, okay, sure, we deplete basophils, the itch goes away, that's great, but is this really a direct phenomenon, or is it something indirect, or basophils just somewhere along this chain of events, and they're just interrupting? Where in the chain of events are they interrupting? So Feng, my postdoc, she took an incredibly Herculean approach, and she said, okay, let's do this. Let's take control mice and then sensitize mice to OVA. Let's purify the basophils, which presumably harbor the IgE on them, and then challenge them with OVA in vitro, and then harvest the supernatants, and then inject them into mice, just normal mice. And then, in fact, when she did that, the supernatants that came from the sensitized mice significantly evoke more itch from these basophils than from unsensitized mice. Now, she also took it a step further. She took the supernatants as well, and then took dorsal root ganglion neurons out of calcium reporter PERT GCAM mice, put them into a dish, and then did calcium imaging. And in fact, if she puts the supernatants on, we see that actually unsensitized supernatants can activate some of these neurons. But the difference was that the sensitized supernatants activate more of these neurons. And in fact, that's enumerated for you here on the right. So there's something coming out of these basophils that is causing many more of these neurons to activate, these sensory neurons to activate, indicating that there may actually be a basophil neuronal axis here. So we wanted to see, no one's actually had shown that basophils actually interact with neurons at all. So we wanted to see this. Again, they're in circulation. So we then looked in the skin that we were going to inject with OVA, and this was in collaboration with Mark Miller, and we used actually dual reporter mice. So in green, we see basophils, in red, we see sensory neurons. And then what we do is if we actually image this, we see these basophils before we challenge with the OVA. They're there, some of them are moving a little bit, they're pretty static and they're small and they're quiet. But if you go on the right, after injection with OVA, you see these huge activated basophils here, and so and they're of various degrees of elongation and activation. And what we find is that the motility, these basophils are moving a lot more, and they're moving very rapidly. So they have take on a very different velocity, and they make apparent contacts with these sensory neurons in ways that they don't prior to challenge with the OVA albumin. Okay, so it seems that they do interact. So then the question is, basophils produce a variety of substances: histamine, serotonin, tryptase, biolipids. What could it be? Well, it's interesting. It's amazing how much collaboration helps. I was at the National Institutes of Health visiting Mark Hoon, my colleague, and he showed me this paper that was not published yet. And he said that if he chemogenetically activates mast cells, what happens is 3 factors get produced, shown here, one of them being LTC4. And what happens is that these 3 factors bind a neuron, and in particular LTC4 is very, very, potent at activating a nerve. And I had known, we've been studying basophils for a long time, and I knew, aha, this is it. It's got to be LTC4, because LTC4s are very, very much enriched in basophils. It's one of the key effector molecules for basophils. So we actually mined RNA sequencing data sets comparing skin mast cells to basophils, and we looked at components that pathways for the production of histamine, serotonin, tryptase encoding, and then lipoxygenase for the production of leukotrienes. And in fact, it's the leukotriene pathway that indeed, as I suspected, is most enriched within basophils. So, and in fact, if we look in the serum in mice that are challenged with OVA versus irrelevant allergen BSA, we find that soon after challenge, LTC4 is greatly enriched in the serum. And if we deplete basophils using our diphtheria toxin BASTRAC technology, we find that that you could actually quench away the LTC4. If we do additional calcium imaging, what's turned out very interesting on the neuronal side is that it completely is housed, the LTC4 responsive neurons are completely 100% housed within a dual positive population for capsaicin responsive, so these are TRPV1 positive, and AITC responsive TRPA1 positive neurons. And in fact, if you knock out just TRPV1 or TRPA1, this acute neuronal response is very much, it wiggles around it. In other words, you have to block both to get rid of this. And in fact, you have to actually block both TRPV1 or knock out both TRPV1 and TRPA1 to get rid of this acute itch flare. So then the last kind of question for us is this really neuronal, could this really be neuronal expression of the receptor for LTC4? So LTC4 can bind cis LTR2 on it's one of its receptors. So we used the siRNA knockdown of cis LTR2, and the way we did this is we actually injected intracisternally into the cerebrospinal fluid. This siRNA then communicates into the dorsal root ganglia, and then what you can do then is knock down expression of these receptors within the dorsal root ganglia. How does this look? We buy PGP 9.5 neuronal staining here in red, and in green you see cis LTR2 staining here. If you merge these, you see that there's overlap. If you put in control, if this is with control siRNA, if you actually put in now the siRNA to knock it down, you see that you lose expression of cis LTR2, and that's quantified on the right. So with this intracisternal injection of the siRNA, what we find is that we can actually knock down this acute itch flare as well that we don't see with the controlled scrambled siRNA. So this was a bit of a journey. In the first half of the talk, I talked about how kind of some of our recent discoveries with regard to lymphocytes and cytokines and how they mediate chronic itch in the setting of atopic dermatitis. But how by thinking more deeply about these clinical questions, what we've come to appreciate is how, in fact, itch and acute itch flares may reveal itself and may help us define why atopic dermatitis is, at the end of the day, fundamentally an atopic disorder, as a response to allergens can result in binding of IgE to poised basophils, which are angry and upregulated for the receptor for IgE in the setting of chronic barrier inflammation. This then activates an alternative pathway that's histamine independent, that is enriched for LTC4, which binds at the neuroimmune interface, the cis-LTR2 receptor, to mediate this acute itch flare pattern, So 2 different modules of itch, chronic itch and acute chronic itch, involving very different immunologic and neurologic mechanisms. Does this happen in patients? Well, it's interesting. If you look back in 1982 in the Lancet, Thomas Platts Mills, who I have high regard for, he actually discovered red meat allergy in recent years, had noted that actually patients with atopic dermatitis are heavily reactive in a very acute manner on the skin to allergens. And in fact, what he found was that containing within these acute immediate hypersensitivity reactions, which is not a typical phenomenon for atopic dermatitis, was actually enrichment of basophils. So we think that this actually is something that happens in terms of allergen responsiveness in patients as well. We also think that this has very important implications across many other barrier surfaces, that this is an alternative kind of neuroimmune basophil neuronal circuit. So what I hope it's become clear is that we really think deeply about the clinical problems that plague us in the clinic, and we bring this back into mice to mechanistically address this. We then stumble upon novel mechanisms in mice, we bring it back to validate in humans, and then come back to mice again to see if therapeutically this is going to show promise for clinical trials. And I think hopefully what's been impressed upon you is that we think a lot of the pathways we've looked at and uncovered are very important and are informing a lot of therapeutics. We've been studying one disease very tenaciously for over 10 years. And this, I guess, is a great example how it is worth studying one disease. It can reveal a lot of new biology, both on the immunology side, the neuroscience side, and has really given birth in many ways the biology to one of the most explosive areas of drug development, in at least in my field, and probably allergy as well. I want to, last but not least, thank the people who did the work. I mentioned all the work. It's a real teamwork from my lab, but Landon Etchen, Fang Wang, I want to highlight the lead authors on the work, our wonderful collaborators at WashU, as well as in industry, at other institutions, are the NIH, of course, and other foundations. And last but not least, I want to thank our patients for helping us with a lot of our studies and helping us to understand better human disease. And thank you all for listening.
Hey, Brian, this is Todd Margolis. Thank you so much for joining us and giving us this overview and up-to-date sort of summary of where you are. It's terrific work, and you've gotten a lot of work done very quickly. We're obviously very interested in this for ocular disease. But I'm going to ask you another question, and I'm way out of my depth here. But you talked about atopic disease and talked about them with elevated serum IgE as your model. But I thought that was just a subset of atopic patients, that in fact was the minority of the atopic patients. Do I have that wrong, or help me understand this?
No, you don't have it wrong at all, Todd. I think the big, it's amazing. There's so much lack of clarity about this. I think the big misunderstanding in the field of allergy is that elevated IgE means anything. What really matters is what kind of IgE repertoire do you have and what environmental allergens are you exposed to, right? So you could have skyrocket at IgE of 5,000, but if the IgE is not recognizing anything in your environment because you live in the desert, you'll probably not have a problem. So that's really what it comes down to. So I think there's a lot of false negativity with IgE. So there are a lot of patients with atopic dermatitis have, quote, normal IgE, but we actually don't know what the repertoire of their IgE is. And actually, in fact, if you do look at their IgE repertoire, you'll find that they have a very kind of patterned reactivity of their IgE to, house dust mite, to pollen, to things that healthy individuals would not even react to.
All right, that helps me, because in reading the literature, this was always very confusing to me. Thank you.
It is. It's the specificity of the IgE that matters at the end of the day, and we just haven't really talked about it that way enough.
Yeah, it makes sense.
Brian, a really excellent presentation. I see why you've been so busy over the past few years. Two questions. Number one, recently benralizumab has been apparently approved for the treatment of atopic dermatitis, and its target, I believe, is IL-5, which was different than the targets for dupilumab, which, as you say, were IL-4 and IL-13 that were recognized. Do you know what the rationale is for the responsiveness of atopic dermatitis to Fasenra or benralizumab?
Yeah. So I don't think actually the IL-5 antagonists, including benralizumab, is approved for atopic dermatitis. It is approved for a number of conditions, hyper IgE, which can manifest with atopic dermatitis, but I don't think it's formally FDA approved for that. Now, would anti-IL-5 antagonism have an effect on atopic dermatitis? I think it certainly could, because these type 2 cytokines kind of feed on each other. So you could potentially use it to disrupt the axis. I don't think that it will go that way because these other agents are so effective. I think you would have much more modest efficacy with IL-5 antagonism. Mm-hmm. I don't think it's really clear in terms of, well, it is clear, but IL-5 tends to go more with kind of eosinophilic disorders, and in disorders in which we think eosinophils are much more pathologic drivers, which is a very fuzzy area. Eosinophils are funny because they'll be there in loads, but it's not clear that what they're doing in a lot of disorders.
Yeah, and so that's why it's actually approved, I guess, for eosinophilic asthma as a treatment.
Exactly.
The second question is, as you know, particularly with acute flare itch, very hot showers can provide dramatic relief. What do you believe is the mechanism by which that occurs?
Yeah, so there are two things that people do to ameliorate their itch that are very homeopathic. Hot showers, which is unfortunate, and I'll say why that's unfortunate. But what it probably does is heat activates TRPV1, and the majority of the nerve fibers are TRPV1 positive. So what you're doing is you're just exhausting the nerves so that they just kind of poop out. So they're tired of doing anything. You could also, the opposite way in which you could actually also help itch, which patients do, is they'll put ice on their skin. And the way that works, it actually works to other TRP channels called TREM8. And what that does is it's why menthol is alleviates itch. The cooling effect actually suppresses itch at the neuronal level as well. The problem with the hot showers is it's very counterproductive because acutely you'll feel better, but what happens is you vasodilate the skin. And you actually cause almost like a body-wide rosacea. And what happens is the lack of vasoregulation is incredibly harmful from an inflammatory standpoint, because vasoregulation is a key component for resolution of inflammation anywhere, but in particular, because you can imagine these are the pipes that are regulating flow of inflammatory cells in the skin, and how you keep them open or closed is very important for resolution of inflammation.
And what is the name of the product that you mentioned avoids that? You mentioned the application of a topical cream.
Well, the cooling, the menthol, just
a mentholated.
So you can
Very
go
good.
creams to try to alleviate the itch. Some might be helpful, some might not be. Some may be even counterproductive. I think it's a dangerous way to alleviate. In the Gulf Coast where there are the no-see-ums, what I've heard is when people get the no-see-um bites, they're very itchy. I got attacked in Florida. What they've learned down there is they take a hot hair dryer and they put it on their lesion to alleviate the itch.
Mm-hmm. Yeah. Well, thank you very much. It's very, very interesting.
Let's see if there's anything in the chat.
Hey, Brian, excellent talk. Can I ask you a quick question related to, you have referred to us, many patients related to the to lipomab treatment. After the treatment, their clinical symptoms of skin allergy is significantly improved. However, they do have some ocular symptoms. Not necessarily itching, but they have a lot of ocular irritations. I'm wondering if you have any insight about the inciting factor, or what's the mechanism of the ocular irritations on those patients?
Yeah, so we have two theories about it. One goes down to the, so I'll give you two, we have two theories. I propose to Regeneron, but they're not particularly interested in mouse work. So I think mouse work's really important if you really want to know what's going on. So basophils bind IgE. Interestingly, what we found is that if you knock out IL-4, what actually happens is that the basophil frequencies go way up in the blood. So I think what could be potentially happening is this kind of homeostatic property of the basophil is lost when you block IL-4. You're able to bind IgE much more robustly. And then that's why maybe you get this kind of conjunctivitis type thing. That's not a good thing. That's one possibility. The other thing I'll go is on the neuronal side is that there are some of these nerve fibers that actually, this is just published days ago in Cell, but I've known about it for a while, is that there are these certain nerve fibers that express MLC that are expressed mast cell function. So they actually counter-regulate mast cells. They express IL-4 receptor alpha, and I think what could happen on the neuronal side is by blocking IL-4 receptor alpha, negative regulation for the nerves, and then the mast cells go crazy. So I think there are two mechanistic possibilities there that we've been entertaining. We're not really pursuing it, just because it's kind of like I don't know. We could pursue it, but it's not really like the big question of the day for us. But if someone would fund it, we would do it. But
sure.
But anyway, so there's some very interesting hypotheses.
Could everybody who is not muted, please mute your screen because we're getting a lot of background.
Yeah, and I have another question regarding your sensitization of the basophil with ovabumine, and then you use the supernatant to challenge the naive mouse, yes. And then can cause. And then do you think if you isolate, just not necessarily have to culture those cells, you essentially adaptive transfer, just transfer those basophil, you have the same sustaining effect on those naive mice?
I think it's possible. I think one of the challenges with basophils a little bit is that they're so low in number, so that there's some technical challenges, and they don't survive very long. So that there's some technical challenges to transfer. But yeah, I believe that if you're able to transfer the basophils in sufficient numbers, that you would recapitulate the biology. And if you look at patients, when you get a CBC, there are patients that have basophilia. They'll have elevated basophils, and they often will have allergic disease.
Thank you.
Oh, I have a quick question, just naive question. So all you talk about those clinical trial, the drugs, are they could make the eye drops and then to treat the eye ocular types?
Definitely, why not? Certainly with the JAK inhibitors. I don't see why not, because that's a small molecule. I think the issue with a lot of them are monoclonal antibodies, so I think that's where you're going to run into a more challenging issue. But yeah, I think that I think they could be used as eye drops. And I think the other thing that's really interesting about the biology and a lot of the work that Chin Lu's doing is that the neuronal composition around the eye is different. So the composition of those nerves are different. So the inflammatory and itch physiology in some of these mediators, I think, are going to be quite different in the eye as well. So I think there's a lot of kind of new biology that's much more directed at that site that could be unveiled, and hopefully, that will happen in the coming years. So I think there could be therapeutics that are a little bit more tailored.
Cool. So if there's no more question, thank you for coming to give this wonderful seminar. And good luck with all the your research. And thank you again. And so I'll put this as a big applause reaction.
Thanks for having me, everybody. I enjoy talking to everybody.
Okay, thank you. Bye.